Hi, my name is Connor Eckerment, and I am a sophomore at the University of Mount Union. And this presentation today will be about the history of the discovery of DNA. Now, start off, what is DNA? DNA is one of the essential building blocks in our body, and it has functions like, for example, it carries our genetic code, and it carries instruction for the creation of proteins in our bodies. And DNA is made up of nucleotides, which are basically what makes up each part of the DNA. And these nucleotides are made out of deoxyribose sugar, a phosphate group, and one of four nitrogenous bases: adenine, thymine, guanine, and cytosine, commonly known as A, T, G, and C because that's how they're shortened when talking about DNA, because the sugar and the phosphate make up the outer part of and then the bases make the inner part of the DNA, which there have to be specific pairs. For example, A and T have to be together, and G and C have to be together. Because it can't be it's like A can't be with G and T can't be with C because they have to be with their partners. Or else DNA would be malfunctioning and better. For example, basically that would look more like a genetic defect. So now, Frederick Meister, and I apologize if I pronounce his name wrong, but he discovered DNA. The first person to discover DNA, and that was in 1869, and he called DNA nuclein. And it was found in the nuclei of human white blood cells. And he found it by isolating the white blood cells from used bandages in order to figure out what it was. And with that, he then extracted the many proteins from the cells, and noticed that one protein in particular didn't match. It didn't fit the rest of the protein cycles. So he named a nucleon, like I mentioned, which became known as nucleic acid, and then later deoxyribonucleic acid, DNA. So with that, we get to Friedrich Levine, who had multiple discoveries about DNA and RNA. And for example, he figured out the three components of nucleotides. He figured out the carbohydrates in DNA and RNA, as well as discovering how they function. I believe that was him. Then we get to Oswald Avery, who wanted to discover what the transformer hormone principle was that was produced by pneumococcal bacteria, which he found that the transforming principle was DNA. And he and his partners at that time had thought that maybe that this discovery might be applicable to other organisms, which proved to be correct in the future. As we go along, the rules of DNA with Irwin Chargaff, he studied DNA in different species to try to find differences between them. And what he found was that nucleotide composition in DNA is different in different species. He also found that all DNA, no matter what organism or tissue type it comes from, maintains certain characteristics, even as its composition varies. So DNA can be different than in humans. As an example, DNA can be different in humans than other animals, but the DNA characteristics are always the same. And these findings became known as Chargaff's rules. We get forward, if you have the DNA structure discovery, now we get more into that with Rosalind Franklin, who started her scientific career in 1947 through 1950s, the first few years after she graduated college. She worked at the Laboratoire Central des Services Chimiques de l'État in Paris, which I know I completely mispronounced that, but she learned techniques of X-ray diffraction, which she later used to take pictures of substances to identify the structure of DNA. Which she did that at the future place of work, which is a lab at King's College. was also where Maurice Wilkins worked eventually, but originally started off working as a nuclear physicist on the Manhattan Project in the 1940s, which was responsible for the atomic bombs that were dropped on Japan at the end of World War II. But he was displeased by that, with what they were made for, so he eventually switched to doing stuff with biophysics, working in the same lab as Rosalind Franklin, which when he got there, he originally studied genetic effects of ultrasonics, but then he decided to switch to studying ultraviolet microspectrophotometry of nucleic acids in cells, which he used that to use his future studies that were kind of similar to Rosalind Franklin's to try to figure out the structure of DNA. Then their work was then used by Francis Crick and James Watson. Now Watson was a zoologist, and Crick was a physicist. And they had met at the Cavendish Laboratory, where Crick at the time was doing one of many X-ray crystallographic projects, because in that lab at the time, they were doing multiple of those projects to try to figure out how different things worked. And when they were there, they decided to work together to make a model of DNA to find out its structure, which eventually turned out to be successful. Now, how it was made is they used known molecular distances and bond angles to create cardboard cutouts of the 4 bases and the other subunits of nucleotides, which were the sugars and the phosphate groups. And with those, they shifted them around on their desktops to try to fit them together like kind of like puzzle pieces were. But however, along the way, they did make some errors and they had to redo some cutouts and fix some things, but eventually they got to everything fit perfectly, that everything fit right. So they were able to figure out what the structure was and how it worked. And they call that the double helix model of DNA. It's the basic structure of DNA. Although DNA can change, I should mention DNA can change its shape, more complex shapes. And DNA can actually be up to 6 feet long as well. In the future here, how is DNA used today? So DNA is used in many different things, like for example, crime scene analysis, like identifying the victim, substances, etc. Ancestry DNA testing, which can be used to find out where you came from, maybe find out more about your family, like your genetic traits, etc., stuff like that. And also like genetic genealogy, which can be used to identify like the family tree of the DNA of the human DNA of the individual the criminal justice that they're trying to find, which actually has led to many different killers, like for example the Golden State Killer, which I'll talk about in a moment as we get to the use of DNA in criminal investigation. DNA is used in criminal investigation in many different ways. For example, like human identification, drug identification. Human identification, with that, DNA can be used to like identify like human remains found somewhere, to be used to identify like a missing person or like a wanted criminal, to track them down, stuff like that. It could be used for other stuff like that as well. With this, detectives have been able to use so many court cases, like the Golden State Killer with genetic genealogy, which is used nowadays in different criminal procedures, depending on if it's needed or not. With that, you can also use it for like drug identification. For example, a good example of that would be marijuana. Like a lot of plant-based things, because plants have their own set of DNA, and with that, nowadays, they're able to the scientists working in labs are able to figure out, okay, what is this plant-based? Is this marijuana? Is it not? Like, is it this or is it that? Like, they're able to figure out what this is, which is able to solve crimes easier. Now, as we go forward here, just a little conclusion here. DNA is an essential building block in our bodies that contains many important functions and other things as well. DNA was originally discovered in 1869 by Frederick Meister, and all the DNA has occurred over the years, including the discovery of its structure. And DNA is used for many different things like crime scene analysis, ancestry testing, and genetic genealogy. With that, it's our works cited page. And I hope you enjoyed this presentation, and I thank you for listening.